Okay, continuing on. You'll start to get practice of trying to identify different phases of meiosis. And the hints I kind of gave you was, well, the general thing is if you see one cell becoming 2, you're in meiosis 1, 2 cells becoming 4, you're in meiosis 2. So take a minute, just in your notes, just see if you can identify 1 through 8. Go ahead and talk it out with your neighbor if you want. Identify 1 through 8. In your notes. All right, I think that was enough time. Just making copies while you're doing that. But anyways, number 1, what'd you put? Telophase. 1. Number 2, I see 2 cells lined up, metaphase 2, telophase 2, metaphase 1, anaphase 1 separating along these chromosomes, prophase 1. I see 2 cells. That looks like prophase 2. 2 cells becoming 4. This is anaphase 2. All right. We have a model set of this. Is over there. You can't see it's behind the cabinet depending on where you're sitting. It looks like this. And they don't have all the steps, but they have some of them. And one thing I like about this model set, I labeled it for you, is they just kind of like have various phases of interphase all the way to prophase, early and late. Okay. And something I like to point out that I couldn't on a previous figure was the separation of the centrosomes. Here, there's one, not replicated. There it is. Okay, it's replicated, and then they start to pull apart. Okay, and they do have some crossover events illustrated there. And they go from metaphase 1 to anaphase 1 to telophase 1, and they only have 2 steps for the telophase for meiosis 2 there. But it's right over there. Meiosis is part of the deal for this next unit exam. And more importantly, meiosis is involved in both spermatogenesis and the ovarian cycle shown here, and that's what I want to do next. So we have all of these structures with oocytes undergoing some phase of meiosis, and I want expectation for students is to be able to track which phase of meiosis you're in throughout the ovarian cycle. So what you want to do is correlate your notes in a way where on one side, keep track of meiotic events. And on the other end, you're keeping track of what's happening in the ovary. And we did talk about this in endocrine. To review that, what we had discussed was the only cells that actually undergo meiosis are the oogonium. So let's go before birth for a female. Oogonium. Oogonium. is singular. It ends with an A. It's plural. But let's say you have one cell. They're gonna undergo mitosis first, not meiosis, where one cell becomes 2 and so forth. Regular cell division to proliferate the number of germline cells, the eggs, the oogonia. And I said that at 7th month pregnancy, you get the formation of a primordial follicle. Primordial follicles form. And I described the structure of that already. You can go back and review that if you need to. And maybe you get something like 1 to 2 million of these follicles. And each of these follicles has an oocyte in it. Maybe that's an oocyte. What I drew in black is the oocyte, the egg, and it's surrounded by follicle cells. If it's a primordial follicle, the cells are more or less flat. This is what I drew before. However, what you should know is that in meiosis, the nucleus, they're kind of in prophase 1 arrest. They're not going to progress past it. That's what's happening inside the primordial follicles. And when you're born, skip to birth. Yeah, so you have something like 1 million primordial follicles. And they're all in prophase 1 arrest. Sexually immature, right? You're just born. And well, eventually you have to get to the part where you're mature enough to cycle through. And so when you reach puberty, you might be down to something like 400,000 primordial follicles. There's some attrition. So let's get from birth to puberty. About 12.5 years. You're still in prophase 1 arrest. And maybe you're down to, like I said, 400,000 primordial follicles. You should be referring to the oocyte as a primary oocyte. Each follicle contains a primary oocyte. Primary because you're in prophase 1, you're in meiosis 1. At any point that you're in meiosis 1, they refer to the oocyte as primary first. And that's about all you can say. Now, when you reach puberty, that's kind of the tipping point where you'll start to go through your first menstrual cycle. And so what you want to do is select a cohort of 20 follicles to develop. Now there's enough FSH, LH to kick in. When you develop the follicles, that's called the follicle phase of the ovarian cycle, the first couple of weeks. So the follicular phase roughly 2 weeks of a 28-day cycle. You'll see the phases that we talked about before. The primordials are shown here. I think that's the next slide. Yeah. You can see the histology that we talked about. You can see, well, the picture, these are the primordials. That's a primary. They stick it in with the primordials there. And it's the small immature follicles that are arrested in prophase 1. And let's remember, I kind of like what I put on the slide there. That's 23 tetrads, right? And if you want to kind of label it, it's 2NR. It's diploid replicated. When you progress to primary follicle, you know what that looks like? We studied it under the microscope. In terms of meiosis, same, nothing's changed. It's still an immature follicle with a primary oocyte inside. Prophase 1 arrest. 23 tetrads. Call the follicle with the oocyte a primary oocyte inside. So the primary follicle, not to be confused with the thing that's inside it, primary oocyte. The follicle, there's many follicle cells surrounding the one cell. Okay. Common mistake in physiology is students start calling the primary follicle a primary cell. Now, why is that incorrect? Is it a cell? It's not a cell. 1, 2, 3, 4, 5, 6, 7, 8. Should I count all the cells? I mean, of course not. I'm just illustrating the point. Pardon me for descending into sarcasm, but there's an oocyte. That's a cell surrounded by many cells. So call it a follicle. Calling it a cell would be incorrect. It's an immature cell. It's not really have the equipment to make estrogens. But if you progress past this phase, you get a more mature follicle. This is like an early secondary follicle. So now we're talking about the 20 that are making it past primordial to primary. Okay? And of those 20, how many will ovulate?
1.
1. What if all 20 ovulated? Not enough room in the uterus. I mean, if you have twins, you know that the birth weight goes down, right? It's like, normal birth weight should be like 7 pounds-ish. Normal birth weight for twins is like 5 pounds or less. In triplets, quadruplets, the birth weight keeps going down. There's only so much room in the womb. But, they can do remarkable things in the NICU, even with a low birth rate. But the uterus is designed for 1. Okay. So typically we say there's 1 ovulation. When you progress, you get the 20 follicles that kind of reach this secondary follicle phase. 20 reach the secondary follicle phase. This is the more mature follicle that has the structures that we associate with follicles that are mature. Okay, note the ZP and the zona pellucida and the theca. Okay. So if you have the nucleus with the chromosomes in it, there's the nucleus with the chromosomes in it, buddy up. And here's the cell with the cytoplasm. Maybe that's just the oocyte. Call it a primary oocyte. Around that would be a zona pellucida. It's like a glycoprotein eggshell. There's other purposes for it. It has sperm receptors. So I just kind of drew just zona pellucida around the nucleus. This is the primary oocyte. I already taught granulosa cells and theca. Okay, I didn't label granulosa cells here. Maybe I should have. We have many layers. I'll just draw it as red lines. Granulosa cells. And around that, you have connective tissue layers called the theca folliculi. Theca for short. And the physiology that you had to know for the last test is still on this test. The granulosa cells, well, I'll start with theca. The theca cells secrete androstenedione. And that is transferred to the granulosa cells where aromatase converts it to estrone. This is all under the influence of FSH. Aromatase converts androstenedione to estrone. So I'll write that as a review. You should still know that. Retain that information. Well, anyways, this is why it's a more mature follicle, because these cells are doing the work, they're hormone responsive, they're responding to FSH, and you're able to secrete estrone, which will develop the endometrium. So these are the more mature follicles. You're pretty much still in prophase 1 arrest, all 20 of these, but not for long. Tetrads. In each of those, what we call a primary OO site. So I'm going to move on. Okay, of the 20, one will kind of progress beyond secondary. Here's late secondary. So what I say is one of the 20 is selected to undergo further maturation. There can only be one, usually in a typical cycle. Primary oocyte of this secondary follicle, it completes meiosis 1, ejects what's called the first polar body containing 23 dyads. That's basically the end of meiosis 1. So select one of the 20. In this round, select one of 20 to mature further. For example, into late secondary. The other 19 are still useful for secreting estrogens during the follicular phase, but you see some more development there. There's more fluid-filled spaces. Okay, I still put ZP and PICA. And so, one follicle completes meiosis 1. I guess how I've been drawing it, if I drew, for example, just the cells pinching off, one cell becomes 2. It's unequal division of cytoplasm. When you finish pinching off, the unequal division, all the cytoplasm goes to the thing that will be ovulated. Okay? This is called a polar body. It's the first one. Basically, it's a cell that got no cytoplasm. You just ejected the genetic material in it. In each cell, when you're done finishing off the split, and it finishes, that cell's got 23 dyads, polar body. It's got 23 dyads. When you complete meiosis 1, each of these structures, first polar body, it's NR. It's haploid replicated. So is this one, haploid replicated. But once you kind of like go into meiosis 2, well, let's stop there for a second. Any questions on that first division? We did a cell division, unequal division of cytoplasm. Now we have 2 cells now that go into meiosis 2. First polar body, secondary oocyte. That's what you call this cell. This one. Why? Because it's entering meiosis 2, secondary. And this one is in a follicle that's going to go from late secondary to Graafian. Okay. So for ovary, excuse me, I will write one follicle reaches full maturation, graft. One follicle reaches full maturity, grafting follicle. So we have our first meiosis one finishing, our first split. So I'm just going to draw the oocyte. I'm not going to draw the whole grafting follicle. You've got a moon-shaped antrum. So there's the genetic material inside there. Okay. So what we got is, I'm going to draw it a little different than they do. Here's my secondary oocyte. It's NR, secondary oocyte. Here's my PB. Got no cytoplasm, just the genetic material. It's also NR for our discussion's sake. But let me draw the zona pellucida around it, because they're all stuck inside the zona pellucida. CP. First, polar body, which is also haploid replicated. So that's what we got. And we enter meiosis 2 when you get to basically the Graafian follicle stage. But you kind of like stop. Let me read what I have on the slide. Meiosis 1 completed. Now the oocyte is now a secondary oocyte. The secondary oocyte begins meiosis 2, but arrests in metaphase 2. Metaphase 2 or anaphase 2, depending on which book you read. Okay? So the chromosomes align, but it's like this. There are cells in the follicle that communicate with the oocyte saying, don't finish meiosis 2. You need sperm to be present. Okay? There's no point in finishing, there's no sperm to fertilize. So you see metaphase 2 or anaphase 2 arrest. Could be both, could be sometimes one, sometimes the other. The point is, don't complete meiosis 2 until sperm is present. That's really the point. Okay. So you're in Graafian follicle, and you're ready to be ovulated. We're talking about day 14 of a 28-day cycle. Ovulation is about day 14. And the hormone LH will kind of induce it. We'll talk more about that later. Here's a picture of it there. Bursting out, rupturing through the wall of the ovary. And hopefully, what will catch it? Uterine tube, right? So that structure is ovulated. Along with all the other stuff, the ZP, the polar body, it's all ovulated. So ovulation, it basically marks the end of the follicular phase. Ovulation of Graafian follicle marks end of follicle phase for this cycle. Remember, I'll put day 14 in parentheses if you've forgotten that. Because there are things that happen now. This Graafian follicle, just the one, it ruptures, it bleeds, and they call it a different thing when it bleeds. The Graafian follicle is ruptured, And it's called the corpus hemorrhagicum. Let me write that down. Corpus hemorrhagicum. I think I got that right. Hemorrhage. It's just a bleeding body. I won't. My feelings won't be hurt if you just call it a bleeding body. Hemorrhagicum is kind of a handle. That's what it means. Because that ruptured follicle, it has been ruptured along with the ovary. It bleeds, and it is actually transformed into something called the corpus luteum. Well, you already know what that is. It's supposed to be on this morning's quiz. Here's a picture of ovulation, though, for real. Okay, and the previous one was for real too. But I like this picture because it kind of shows it in stages. It's a kind of a real-time view of the follicle, the ovary, the egg. You can see the surgical instrument and frame there, but yeah, that's exactly what it looks like. They must have been doing a surgical procedure, and it just happened to catch the ovulation happening at that moment. So, glad I got that in the slides. The luteal phase begins after the bleeding body becomes the yellow body. This has been ovulated. It's in anaphase 2 arrest, metaphase 2 arrest, awaiting sperm. So assume this awaits sperm. This is now in the uterine tube. Because now we're talking past day 14, post-ovulation. I think that was my first receives, right, when I gave that overview. The uterine tube receives the ovulated oocyte. Now you know it's a secondary oocyte awaiting sperm. But for the ovary, the bleeding body becomes a yellow body. Now I would expect you to write corpus luteum. Luteal means yellow, so that's an important word to know. Okay. And this structure secretes progesterone, and it'll help get the endometrium ready for pregnancy. So there's some pictures of the corpus luteum, including like one I took last year. Looks similar to the one from the publisher. It's just a ruptured follicle that bled, now became something else. And it becomes a yellow body, secretes progesterone for a couple of weeks. Okay, now if no pregnancy occurs, if an implantation does not occur, it will die and it become a white body, corpus albicans, if no pregnancy. So the corpus luteum dies. Becomes a white scar. Literally call it corpus albicans. There's a picture very similar to the one I took for your quiz. Fibrotic scar tissue is what you're seeing there. Of course, we have to talk about if sperm is present, fertilization happens. So that's if not pregnant. What if pregnant? Well, after all, this is the reproduction chapter. Okay? Well, what we could say is that you're going to have a completion of meiosis 2. So let me redraw this. Sperm present for fertilization. Complete meiosis II. So before we had one cell and one polar body. So these two things will become four things. Pretty much the big one will just stay big and you'll get three small ones. Okay? You're going to eject half of this material. So what does that look like? So it's like, if this moves over here. Oops, wrong color. Okay, I just went from dyad to single chromosome, right? And what you did was you ejected a second PD. Second polar body. So that's a structure. This is the secondary oocyte. It's like sperm's gonna get there. Rocket-shaped sperm. That first polar body, I really don't care about the polar bodies. They're not important physiologically. But that could like divide into two. Okay, so these two came from the first. From the first PB. And they're all in the ZP, zone of Follicula. Still there. Okay. But sperm will fertilize the secondary oocyte. That's still what you pretty much call it. Sometimes I abbreviate secondary too with a little circle there, secondary oocyte. Well, anyways, pretend the sperm has, don't pretend, sperm do have DNA. So we got it. Okay, and it's going to contribute, and they're going to like restore the full complement of human DNA. That guy and this is N haploid. By the way, each of the polar bodies are also haploid, right? But I don't care about those. They're not the ones that fertilize and by the sperm, that they don't do anything. So anyways, what we did today was if you track through your notes and follow this figure, what you should be able to do is go on one side, my cell division things, the other side, things in the ovary, just like I talked about as a review. I started before birth, right? And they said there's mitosis. There's just the oogonium undergoing regular cell divisions. However, you do get the primordial follicles form at about 7th month. Okay. And then you get birth along the line. This says infancy and childhood. So you're born, you still have primordial follicles. What I don't, it's okay that they put 2N, but what is it really? It's 23 tetrads. And I didn't call it 2N. What did we call it this morning? 2NR. It's diploid, but it's replicated. So that's one difference that I like to make. I think it makes it a little less confusing. Well, anyways, each month from puberty to menopause. Puberty is about 12.5 years. Menopause is about 50.5 years. So every month, menses means month, you cycle through. Now, how many do you pick? Each cohort, each cycle? Something like 20. And they just illustrate one. You progress through, okay? And all the time, you're stuck in prophase 1. But how many make it to here? 1. And that one is selected to complete meiosis 1. Completed, by one primary oocyte, and basically I'll talk about the LA surge later. But they show one cell becoming 2. One cell kept all the cytoplasm. That's the secondary oocyte. That's the first polar body. Okay. Well, that is the structure that is ovulated, and then you get the corpus luteum, and it will degenerate. However, it will not degenerate if sperm is present. Meiosis 2 completed only if sperm penetration occurs. And so they show all the ends on the bottom. Now, how many ends do you see here?
4.
I see 4. Those 4 cells. Well, these are just polar bodies. They're not true cells. They got no cytoplasm. Okay? You kept all the cytoplasm in the cell that's going to be fertilized. All right? So, yeah, that's where we stop for today. And what we'll do next time, I'll start to talk about ovarian and uterine cycles. And if you want to give yourself a preview, this will take me about an hour to go through, this one figure. Okay. And to give you a preview, this one figure is your essay question for lecture exam 2. Okay. All right. And before you start groaning, students always tell me, "Oh, take him during the summer. He's easier." And I have no idea why students say that. Maybe you ought to start spreading different rumors. Well, what I'll do is I'll talk about the uterine cycle, and I'll emphasize the hormones and how they drive everything through the ovarian cycle and how it's synced with the uterine cycle. I'll map it all on the board, and you'll have a full understanding of female repro physiology. All right, for lab today, some time left for lab.